All right. Can you all hear me? Hello. Good morning, everyone. Also from my side, it's a really extreme pleasure and honor to be here, and I'm absolutely grateful for Valerie. I think she just left the room for really giving so much energy, love, and heart to this foundation and to make this all a reality, and also for Juliette for pushing this field from the very beginning. And we're very fortunate to have interacted also now over the past few years, and I hope this will intensify. And it's obviously an amazing honor and pleasure to meet all of you, and I hope we can interact in the afternoon and speak a little bit, and so you can hear what we are doing, and we would more than love to hear what you're doing, what are the most important questions that you have, so that we from the basic research side can try to help answer those questions with the utmost importance. As you've heard from the nice introduction from Valerie and Juliette, there's not one MID1-like patient. We have a tremendous amount of amazing individuals that make up this community, and we try to understand and help everyone a little bit. And also we have an amazing team of partners in Heidelberg that try to solve this puzzle and try to piece it together from a also biological and scientific side. And along with me here in Rouen is Maya Hempel, who you see here on the picture and who's just walking here. So she's our partner in the genetic polyclinic in Heidelberg that very closely is also interested in anything that's related to MID1-like. And also here is a PhD student from my lab, Jule, also here in the first row. So if you have a chance to interact with her, she's really also studying the molecular basis of MID1-like syndrome. So our research really is basically focused on a very simple questions. And that is also what was introduced by Juliette, that all of our cells have this amazing capacity to form all cells of the body. And these cells, as you know, are stem cells, and they can form, obviously they can form the brain cells in our body, they can form the muscle cells in our body and all the other cells. And in order to do so, all those cells have within their nucleus the genetic makeup or the blueprint or recipe book, as Juliette mentioned, that allows basically all these instructions to be read and to make the cells what they're supposed to do. And in order to do so, it is very important that those instructions are read correctly. And for this, there's what we call transcription factors, and those are genetic switches, proteins that go into this recipe book, that's encyclopedia, and finds the right pages, turns them up, and makes sure those are only read. And normally that happens very nicely. Otherwise, the wonder of life, and we all wouldn't be sitting here because there's so many pages that have to be read accurately. However, sometimes this process can also go wrong, and it has to be tightly controlled. And one such very simple image, if you think about it, is that the book of life has instructions for all cells in our body, right? The brain cells that are making us think, that making us feel emotions. But there's also the instructions for the other cell types. And those have to be obviously shut down. And one of our basic findings was that the transcription factor, so MID1-like, the gene that we are all very interested in, one of its function is to shut down genes, to basically make sure those pages are not opened and read and potentially cause the wrong recipe book to be cooked or the wrong meal to be cooked. This is obviously very simplified, but again, just in a very simplified image, thinking about this, we believe that another analogy to this orchestrator, MID1-like is like a guardian, like a temple guardian that prevents unwanted instructions to be read. And we have studied that for a long time in different contexts, and only recently have also been started to study that in disease, because we have become aware of the work from Juliette and others that there is obviously a potential link between variants in this gene and disease cause. And together with actually very almost simultaneously with the team of Joe Dougherty in the US, we also studied that both in a mouse model and in a human model. And I just want to give you a little bit of an explanation what we do, because it might seem very abstract for some of you. So we want to study what happens if this, what we think is a guardian, is lost in cells. And there are many ways to do so. And obviously, Juliette mentioned where this gene plays a role is in our brain cells. And we cannot easily get brain cells from patients, as you can imagine. But we have now in biological research find ways to generate brain cells in the culture dish, yes. And what we can do in that is, in a very simplified manner, we can take the cells that can do everything, those stem cells, and using genetic tools, such as many of you have heard, this genetic scissors, and change genes. In this case, we made changes or variants that mimic those changes that are also found in patients. And once we do so, we can turn those cells into the brain cells in the culture dish. So this is really a very simplified cartoon. This is a process that takes several weeks and a lot of skills in the lab to achieve. But what it will give you in the matter of a few weeks is this beautiful looking human brain cells in the culture dish, in which we can now study what is happening when this gene, MIT1-like, is changed. And really, I don't go into too much detail, but I just want to give you some of the concepts of what we have found. And one of those is that we started to study the function of those brain cells in the culture dish. And again, just for you to understand how this looks, this is one of those plates which we use to grow these human brain cells on in the lab. And as you can see, there's 48 of these little compartments in which we can grow in the cells, and we can monitor there now the electric activity. As you know, one of the key features of brain cells is to communicate with each other and to exchange electric signals using chemical synapses. And this is how this looks in real life. You have now these electrodes, and on top you see hundreds of thousands of human neurons that kind of lie on that chip that we use in the dish, in the laboratory. And this can pick up the electric activity by monitoring live what those neurons are doing. And so we can look at, are they active, are they not active, how active are they? And I just show you now a little movie how we compare now cells that have normal levels of MIT1-like on the left, and how that looks once we have changed, introduced the variants that are also found in patients on the right here, and shown in teal. So each and every of those light sparks that you will see is the activity of hundreds of thousands of neurons that we can monitor live. Again, human cells in which we change now this protein. And it is always remarkable for us in the lab to really see this process live, because we can now see neurons, human brain cells live at work. And I think it's also very clear to you that there's a striking difference between those two conditions. So once we have less of this variant of MIT1-like, once we have this variant that is disease associated, we can see a severe hyperactivity. We sometimes refer to that like a little thunderstorm in the culture dish, because there's too much action going on what a normal neuron would do. And that might maybe relate to some of the experiences you have also with your children. We also then try to translate those findings from this culture dish to a mouse model. And the reason why we do this, because here we can study more complex processes, how the brain develops. Is there anatomical changes? We heard about that there might be micro and macroencephaly is reported in patients. And those features we can also see in Recapitulate. But what was very important to us is that this also allows us now to study behavior. And one very simple assay how to look at behavior in mice is just to look at them and see how they behave in an unperturbed situation. And if you now, for example, look at a mouse that has normal levels of MIT1-like, this would run around in the box and just explore the box and, however, stay a little bit to the side, is a bit afraid and not too active. When we now look at mice in which we have artificially also changed this gene with MIT1-like, we can see that they are hyperactive. They run around much more than their control mice, and they have almost 70% increased activity, which is also maybe features that can some of you relate to, seen in some of your children. I don't go into much more of those details. We did also, and so did the lab of Joe Doherty, some more tests and assays. One thing I just want to share also with you, however, what we observed is when we looked at the social interaction of those mice, we also observed there was decreased social interest in the mice that had decreased levels of MID1-like, suggesting really that complex behaviors such as social interaction are changed, really just molecularly caused by removing this what we call neuronal safeguard. Really for the first time showing that this could be the cause of the symptoms that we see also translated in patients. So with this knowledge in mind, just again, as a very high level overview of what we observe, because it's all published, and I'm more than happy to tell you a little bit more about the details in the coffee break. What we saw is basically two things. One of the effects when we have a variant of MID1-like that is associated with disease is that neurons are less good in becoming neurons. So there is a neurodevelopmental phenotype also on the molecular level. And the other phenotype that for us at least was very surprising is that even once the neurons have formed, so the development has occurred, there was a hyperactivity of those cells. And that for us is very striking because there might be phenotypes that are not only occurring in development, but there are maybe phenotypes or symptoms that are later in life that could be targeted with therapeutic intervention. And one such mechanism we tried to tackle because of this, what I told you beforehand, observed hyperactivity. We also could relate it back to a specific potential molecular mechanism because we saw that some channels that are important to make neurons active are increased in those cells. And that is very nice because this allows to potentially use drugs that are used in other diseases such as epilepsy to reduce such activity, to block those channels and decrease hyperactivity. And this is exactly what we tried also first in our mouse models. And as you can see, this is again just now the mouse that has a variant of MID1-like that is hyperactive. And when we acutely applied this drug, lamotrigine, that is an approved anti-epileptic drug, to the mice, we could also reduce this observed hyperactivity. Also, what is not published, but what we have shown now is that we can also increase the decreased social interaction once we apply this drug acutely. Importantly, we now wanted to take that back, at least on the molecular level, to the patients. Because what I've showed you is all so far only in artificially generated mouse or cell models in which we introduced variants of the gene that are associated with disease. So due to our research and the really nice network of patients that we have been really fortunate to interact, we also now we're taking a different approach. And that is a very, again, exciting approach in biology in which we can now take the blood of patients, turn this blood into this cells that have this fantastic ability to become any type of cell in the body, which is a pluripotent stem cell, and in this case, turn it obviously into a neuron, which is the cell type that we are mostly interested in because it is affected by MID1-like syndrome. So just again, also maybe catching back on what Juliette said, one of the questions that we were really interested in was the idea that there are different variants occurring and reported in patients with MID1-like syndrome. And one is what Juliette also said, some letters are changed, which is missense mutations. So just a single part of this huge protein has one tiny change. While the other big family of changes that occur in patients are changes that lead the protein to be either truncated or completely degraded. And these two, as Juliette said, seem to clinically not have a significant difference. And we also tried to understand this now in actual patient cells. And we were very fortunate, as I told you, to really have now also a nice interaction and cohort of patients that are also excited about this basic research and try to use this knowledge to further our understanding of the disease. And we first wanted to test if now in cells that we get from the healthy family members compared to the children, we see any changes that we observed in this artificially engineered cells. And as you can see, once we test this in cells that really have this loss or decreased level of MID1-like, we see the same hyperactivity that we observed in the engineered cells, which makes sense, but it's also very nice to confirm in actual patient cells. And importantly, also those patient cells, really important to state, in culture react to the acute drug treatment with lamotrigine. And then for us, the very important experiment was, as you can see here for Jule, performing those experiments in the lab to test if now this other big family of changes or variants, which is this one changed letter, could have similar effects. Again, when we compared healthy family member cells to cells in which a change in one of those letters occurred, we observed the same hyperactivity, which is in line with the clinical features that Juliette mentioned. And also here, we can, with the acute application of this drug, reduce this hyperactivity. So it's very important to state that these findings are only in preclinical mouse and human cell culture models. We don't know if this has any real-world implications for the patients and the families. There are now several patients in Germany that started to take this medication in collaboration with their treating physicians. However, we have both mixed results. While some anecdotal reports say there is positive effects, several patients also actually stopped the drug because there have been adverse effects. So it's for sure no magic bullet that we have found here. But we're really trying hard to better understand if this could be a treatment for maybe some patients, or understand why in some patients it works while in others it doesn't. So if you or any of your loved ones in your network have taken this medication or are thinking about taking this medication, I would really urge you to get in touch with us, because we need to collect this data to make an informed decision, to make obviously also enable us to give recommendations to families that contact us, because at the moment we are the basic researchers that want to understand this in the culture, but we need you to make clinical impact on that. So with this, actually it is important to state there's many open questions that we are also in the lab trying to understand. And I just have some highlights here, and I'm more, as I mentioned, than excited to interact with you to hear what you think is the most exciting questions. So we want to understand the differences between patients now on the molecular level in more detail, to see if what Juliette is observing in the clinic also holds true really in the culture dish. Also, we want to connect these molecular features with the developmental trajectory, so how the kids and patients develop over time. And this is why these meetings are so important for us as basic researchers, also to see children that are of different age, that have different experiences and different developmental trajectories, and learn from that. So it would be really great to connect with every one of you, basically. As I mentioned, the clinical effect of lamotrigine is for us very important, because we think there might be usefulness, but it's, as I said, definitely not a magic bullet. And last but not least, we're also interested in developing additional strategies to potentially have a therapeutic impact in the disease. And Joe Dougherty's team is also following approaches, and it will be very exciting to connect also with researchers in that area. Just one experiment that I want to show why we think there might be also other strategies that could be really more of a therapeutic approach is that, again, coming back to this hyperactivity phenotype, just a different visualization of that is when you have, in this case now, mouse cells that have normal levels of this, what we call neural safeguard, compared to no, you see this hyperactivity. And when in cells, again, in cultured neurons, in mouse, in vitro, we now apply using, let's say, an in the dish gene therapeutic approach by bringing back this transcription factor. In culture, we can also reduce this hyperactivity, at least suggesting as a proof of principle that regulating the levels of these factors by any means, for example, using viral vectors or antisense oligonucleotides, as Joe Dougherty's team is pushing, could really be a therapeutic future avenue. So with this, I have to thank, obviously, the amazing team that stands behind the work. Jule is here, but there have been some really incredibly talented and dedicated PhD students that worked on this project. Bettina, Jana, Bryce, and with the help of Tim and Sarah in the lab. Plus, obviously, the interaction that we have with our amazing clinical partners. You see their faces and names here, most remarkably, Maya Hempel, that is on this journey with us together. And last but not least, obviously, you and your input and feedback, and we really would like to get in much closer interaction with the families and patients to really help make an impact and make a difference. And with this, I'm at the end. This is the family day we organized last year in October in Heidelberg, and it was an amazing experience. It's great to see some faces from that meeting back here, and I hope we can reconnect many times more in the future. And with this, thank you, and I don't know if there's time for questions. If not, just find me later.